attendees, we are moving on to our next presentation. Dr. Vidya.
Hello everyone. Today I'm going to present a molecular study titled as Assessment of Quality and Quantity of DNA Extracted from Mouthwash Samples. We all are well aware that DNA or deoxyribonucleic acid is a hereditary material, so it's present in almost most of living organisms. And this is the source of most of the studies and most of our knowledge that we know. So basically in clinical medicine, forensic medicine, basic science, gene therapy, and also in molecular epidemiology, biomonitoring, it's used variously in various fields. We are aware that the source for this DNA can be obtained from blood samples, urine, hair samples, etc. One of the samples from where the DNA can be obtained is the buccal cells. Now, these buccal cells are a convenient source of DNA. They provide comparable results with the blood samples, which are basically the go-to source for the DNA. Now, buccal cells can be either obtained from your toothbrushes or your buccal swabs or through the mouthwash samples. Now, when we look at blood samples, though it is the most go-to source for the DNA, it also comes with certain limitations. We are well aware that in pediatric population, obtaining blood samples is quite a challenge. It is an invasive process. In geriatric patients, it is going to be a bit difficult. In certain people wherein they fear the needles, such population also it's quite a task. Needle prick injuries is quite a well-known disadvantage for collection of blood. It is expensive because we need an auxiliary personnel to collect the samples. We have to store the samples, send the samples to a different place for the analysis. And especially when you're doing a large-scale studies or molecular epidemiologic studies, where a huge population is required, it is going to be expensive. So therefore, when we do a large-scale or studies, we need a sample which is more comfortable to use, wherein the results are also equally comparable with that of blood. So therefore, buccal cells is one such source. It is easy to collect. You can get sufficient quantity similar to the blood. It is easy to store and transport. Auxiliary personnel are not required, which is one of the major limitations with that of the blood samples. Because blood samples, we need a well-trained person to collect the samples. But in buccal cell samples, we ourselves can collect the samples. And it simplifies repeated sample collection. Whenever we take blood cell samples, because of certain technical issues, we have to again redo the sampling in blood cells. But in buccal cell samples, it's very easy to collect the samples again, which is an added advantage. So therefore, the aim of the study was to isolate the genomic DNA from buccal cells obtained by mouthwashes. We also wanted to evaluate the quality and the quantity of the DNA extracted and also over the various time intervals. We want to check the various time intervals of evaluation because first thing is that whenever we collect blood samples, we used to do the extraction or we usually freeze them. But when we collect the buccal cell samples, we want to know until what time or temperature, at the room temperatures, that we can isolate the DNA. So we used 10 mouthwash samples was considered. Healthy individuals with no significant oral findings were included in the study. Patients with any significant medical history of systemic illnesses or oral pathological states, developmental disturbances, habits such as smoking, tobacco chewing were excluded because for the known reason that they will certainly show certain changes in the buccal cells. An informed consent was obtained from all the participants. Mouthwash samples were collected in sterile containers by asking them to vigorously rinse their mouths with a 15 ml solution of 3% sucrose for 60 seconds. In order to assess DNA integrity over time, the mouthwash samples were divided into 4 equal parts. Now, these samples, after dividing into 4 equal parts, one part was used for immediate extraction. The other parts were kept at room temperature. They were not freezed or stored in under minus degrees. They were stored at room temperatures and extracted the DNA after a period of 10, 20, and 30 days. All the samples which are objective to analysis of extraction of DNA. The resulting DNA isolator was subjected to spectrophotometry to determine the optical density. And then for furthermore, application of DNA was done by PCR and documented using gel electrophoresis. And here PAX9 gene was used, which is a low molecular weight gene. Coming results, spectrophotometric analysis. When we optical density of various time intervals of samples, we extracted DNA from almost all the samples. But the 30-day samples, the extraction of DNA was a task. Among the 10 samples of the 30-day samples, 6 samples showed a very small pellet, and remaining 4 samples did not show any pellet at all. There was complete absence of it. So due to technical difficulties, this complete group was excluded from the analysis. So only immediate 10 and 20-day samples were considered. So the yield was good in 10-day samples compared to the yield was good in the initial samples compared to the 10 and the 20-day samples. 10-day samples showed a decrease by 16%, while 20-day samples showed a decrease by 22% from the immediate samples. The immediate samples were successfully amplified, while 10 and 20-day samples were unable to show the documentation in agarose gel. Here we had amplified for the PAX9 gene. So here, basically, mouthwash samples are widely used comparatively to the buccal swabs because studies have shown that the yield that you get with mouthwash samples is comparative to that of the 1638 buccal swabs. And also here, we used a sucrose solution, which is more palatable and it is easy to rinse compared to the mouthwash samples which are present in market-wise. Now, amplification of samples was done with PAX9, which is a low molecular weight gene. Here, we have to note that these samples are immediate samples, when they show the amplification here, specific amplification was noted. So all the samples were present in equatorial region and they were farther away from the marker. This marker was a high molecular weight marker, whereas the PAX9 lower gene from which we wanted the specific amplification to happen was a low molecular weight. So we can note that everything was an equatorial placement. Specific amplification was carried out and farther away from the marker, suggesting that the particular application that we wanted was successful to happen. We are well aware that by freezing, DNA activity is not going to be affected. Metabolic activity will be arrested, but it will be viable. But on room temperature, whenever we try to store DNA in room temperature, because of susceptible to instability, decay, degradation, contamination, fragmentation, damage, all these are going to happen. Therefore, the yield of DNA may not be that good when you do on a room temperature. So this could be the reason why the 10 and 20-day samples could not provide the amplification, though the extraction was successful. The entire stretch of the particular sequence that we want to amplify here was PAX9 gene. So that particular sequence could have been cleaved into bits, and therefore this amplification was not possible in the 10 and 20-day samples, though the extraction was successful. So the aim of the study was met as we could successfully isolate DNA from buccal cells obtained from mouthwashes. Moreover, we could determine the optical density and amplification for a particular sequence was possible, which was PAX9 low molecular weight gene here. We do find various studies which are in supportive of the present study. Vidya et al. Found the mouthwash samples produced consistently better results than the brushes. Singh et al. Concluded that they were equivalent for PCR reaction requiring immediate DNA fragmentations, but the mouthwash was far more superior for reactions requiring long fragments. Kaiser et al. Compared cheek swabs and mouthwash for buccal cells collection in the Black woman health study and concluded that the mouthwash was superior in terms of success of PCR and DNA yield. Studies have also shown that mouthwash method has been validated in mass screening with PCR amplifications for specific mutations with specificity and sensitivity of 100%. Our findings were also consistent with Heather Spencer Filson et al. In their study, they showed that 10 and 30-day samples had significant DNA than those processed after 1 day. Also, Hain et al. Showed in their study that DNA could be extracted at different temperatures. And here they have extracted at 25 and 37 degrees also, and for a maximum of 1 week with less of PCR failures. Lum and Lee Merchant et al. Reported that storage of unprocessed samples at room temperature for 1 week did not affect the DNA yield or ability to PCR amplify. Gracia Colors et al. Found that storage at minus 80 degrees centigrade for up to 1 year did not significantly deplete the amount of DNA in the samples. So here we see different temperature ranges which support that until 10 days maximum predominantly was sufficient enough for the DNA to be extracted when stored at room temperatures. But however, in contrast, we found certain studies wherein that Ellen H. et al. And Marcy Ajar, they were not able to extract the DNA for a period of 2 weeks and 30 days respectively. We have to note that here they were able to extract, but they were unable to extract. We have to note that we come from a tropical climate, that is, the study was done in India. So therefore, the lab equipment and the temperature definitely does vary, and therefore this can act as a limitation when we're trying to store the samples. DNA tests are being developed to recover information from smaller regions of DNA, which are more likely to be intact following DNA damage. These new DNA tests include mini SDRs using PCR primers close to the SDR repeat region and single nucleotide polymorphisms. Whole genome amplification and DNA repair methods are also being evaluated to determine the possibility of enriching PCR amplification material from limited or damaged DNA templates. This improved technology will definitely enhance those cleave bits that we had lost in 1080 samples to amplify the DNA in those samples too. Further directions, we were able to extract DNA until the immediate extraction was possible and 10 days extraction was possible, though we saw a dip in the amount of the yield of DNA. So therefore, as further studies are required to evaluate DNA isolation application from immediate to 7 days duration at an everyday interval to arrive at a conclusion for a period of maximum DNA yield. Conclusion, mouthwash samples provide sufficient quantity of DNA which can be utilized for mass biological studies. Therefore, this appears to be a simple procedure which is non-invasive, which is likely to increase participation and decrease costs in molecular biological studies and can be an alternative to blood samples. My references. Thank you.
Dr. Vidya?
Yes.
Now I request the participants to raise queries to Dr. Vidya. Ms. Vidya, we have one question from one of our participants.
Yes. I see. I read that. Is it this removal tissue from a child without the knowledge of the mother for genetic testing for paternity or crime? Yes, it is. DNA material is definitely something which cannot be done without a consent. Therefore, we have to take consent if we're going through legal methods. We do see certain cases coming up, but it is actually extraction of DNA because it has genetic material. It is legally, there is legality possibilities in that.
Any other
Answer?
questions? Yes, ma'am. Yes.
We have to know that DNA is genetic material, and when genetic material is the source for almost the foundation of a living organism, right? So when you are extracting DNA from any possible sources, if the person is aware that it's been done, it's better to always take a consent because legality there is definitely a problem.
We have one more question from Rachel. So what are the legal structures around taking tissues without consent from the person?
The legal structures can be that the person definitely can sue you, the first thing, because most of people, they are Google aware about the importance of genetic material and DNA and what all can be obtained from them. Now, once you have a DNA, the numerous possibilities that you can do with it is enormous. You can alter, you can change, you can do so many things with it. So the vaccines are created, or you can have an alteration. You have the entire genetic makeup of a person. So it is a huge legal problems. It is not something that easy. There's a subject branch in science and legality and the crime sections, everything for that.
Thank you so much, Dr. Vidya. So there's one more question.
Yeah, there are ethical issues. Genetic material differs from person to person, and certain offsprings, certain ethical races will have certain similarities within them. That's the reason we are able to differentiate them into different ethical groups. So all of these will definitely come under legality issues. Without knowledge doing anything or without a consent doing profiling is definitely come under issues. Therefore, you always, when you take a blood sample or any samples, you always give a consent form.
Thank you so much, Doctor.